Chapter 12, Nervous System, Nervous Tissues, Part 5. All right, so when we have resting membrane potential in the neuron, negative 70 millivolts, that means a lot of other types of transmembrane proteins are not active. So the chemically gated or voltage gated channels are all closed, and only the leak channels, sodium potassium leak channels, and the pumps, the sodium potassium ion pump and calcium ion pump, are active. Now, when some of these gated channels open, whether it's bound to a chemical, a neurotransmitter, or because of a change in charge, they then allow ions to move. And the first to open are usually the gated sodium ion channels, allowing sodium ions to flow into the cell, going from high concentration to low, going from the negative outside to the positive outside to the negative inside. And this is referred to as depolarization. So depolarization is making the charge across the membrane more positive. So for instance, the gated sodium ion channels open and maybe it goes from negative 70 millivolts to negative 60. It's more positive. And it's basically making the inside of the neuron more positive compared to what it was before the channel opened. Hyperpolarization is the reverse. Hyperpolarization is making the inside of the cell more negative. This may result when gated potassium channels open, so the potassium floods out of the cell, going from high concentration to low concentration. Or it may be the result of gated chloride ion channels opening, allowing chloride ions to flow into the cell because chloride ions are negative. So when they enter the cell, they make the inside more negative. So either positive is leaving via potassium ions or negative is entering via chloride ions. So you might go from negative 70 millivolts to negative 80 millivolts. That would be hyperpolarized. Now, what kind of transmembrane proteins can open to change the charge across the membrane varies based on what part of it neuron you're talking about. So when we talk about receptive ends, say cell body and dendrites, then we're referring to what are called graded potentials, which are where the change, the depolarization or hyperpolarization, is occurring because of the opening of chemically gated channels, where the neurotransmitter is binding to a receptor, opening those ion channels. And this usually only allows a small amount of ions to cross over, causing a local change in charge. It's more localized. Now, again, this could be depolarization, cations or sodium ion channels coming in, sodium ions coming in, or it could be hyperpolarized, potassium ions leaving, or chloride, negatively charged chloride ions entering. So it could be depolarization or hyperpolarization. If the depolarization is significant enough to move the resting membrane potential up to negative 50 millivolts, then we can get an action potential on the axon. This change is dependent on the amount of stimulus. If enough chemicals, enough neurotransmitters are released, and they open enough channels, say enough chemically gated sodium ion channels, and enough sodium ions enter, then the depolarization will reach threshold to cause the generation of the action potential. However, if it's not enough, then it's only going to be temporary change, and then it will repolarize and return to its normal negative 70 millivolt resting membrane potential. So it can be short-lived and only have no overall effect, or it can be significant enough that it leads to that action potential, reaching that threshold. So you reach threshold, reach negative 55 millivolts. This will now stimulate the voltage-gated sodium and potassium ion channels in the axon hillock. So various ions arrive and lead to a threshold of negative 55 millivolts, and now we get the opening of voltage-gated sodium and potassium ion channels. Sodium ion channels will allow sodium to enter, leading to depolarization, And then that change in charge will be significant, a rather massive amount of depolarization, which will then flow to adjacent areas, causing the next set of voltage-gated sodium ion channels to open, and so on down the axon. So here is, say, the axon hillock and axon at rest, negative 70 millivolts. We have the graded potential from the cell body and dendrites creates a threshold being reached of negative 55 millivolts at the axon hillock. So now the axon hillock has its sodium ion channels, voltage-gated sodium ion channels open. Lots of sodium ions flood in. We get massive depolarization at the axon hillock. This change in charge will then stimulate the neighboring portion of the plasma membrane of the axon. This will cause its voltage-gated sodium ion channels to open. Sodium floods in, gets the massive depolarization, and then stimulates the next region of the plasma membrane to open, and so on, again, so on down the length of the axon. And this is an all or none response. Either you reach threshold and it happens, or you don't reach threshold and it doesn't happen. So, depolarization is followed by repolarization and even hyperpolarization when voltage-gated potassium ion channels open. So first we have the voltage-gated sodium ion channels open, sodium ions flood in, we get depolarization. Then, after a brief pause, the voltage-gated potassium ion channels open. Potassium ions leave the neuron, and this causes repolarization and even hyperpolarization to eventually get back to resting membrane potential. So, after the sodium ions flood in, then we have repolarization. Voltage-gated potassium ion channels open. Potassium ions flood out of the cell, carrying the positive charge out, re-establishing the resting charge of negative 70 millivolts. Sometimes this leads to hyperpolarization, where it goes below negative 70 millivolts before reaching back up to negative 70 millivolts, the resting membrane potential. This could technically also be caused by the movement of chloride ion channels, but we're going to talk primarily about sodium and potassium. Now, because of this, there is what's called a refractory period. Brief period of time when the action potential going down the axon can only go in one direction. It cannot back up and go the other direction. And this is caused primarily by the imbalance in ion distribution. So even though we get the charge reestablished and can return to resting potential, now we have high concentration of sodiums inside, high concentration of potassium ions outside. That distribution prevents a new action potential from being generated. So we have different regions of the neuron where different things can happen depending on the ions and the opening and closing of various channels and pumps. So again, the receptive segments have neurotransmitters binding to receptors, causing chemically gated ion channels to open. If this causes threshold to be reached at the axon hillock, the initial segment, then we get the opening of voltage-gated sodium ion channels. Lots of sodium ions flood in, generating the action potential. That then gets propagated down the axon until it finally reaches the transmissive segments, the synaptic knobs. That then, of course, lead to the opening of voltage-gated calcium ion channels. Calcium ions flood in, and now we get the release of the neurotransmitters to pass the signal on to the next cell. All right, so excitatory neurotransmitters. Excitatory neurotransmitters are the ones that, when they bind, they cause cation channels to open. They cause sodium ions usually to flood in to the cell. So they cause an excitatory postsynaptic potential. So this is the postsynaptic neuron. The neurotransmitters bind, opening the sodium ion channels. Sodium ions flood in, causing a depolarization event, an excitatory postsynaptic potential. Inhibitory neurotransmitters bind and either cause chemically gated potassium ion channels to open and potassium ions to leave the cell, making it more hyperpolarized, or it causes chemically gated chloride ion channels to open. Chloride ions enter the cell, they're negative, so they also lead to hyperpolarization, making it more negative. And this is considered an inhibitory postsynaptic potential. So again, it's affecting the postsynaptic neuron at the synapse. It's becoming more negative, which means it's going to be more difficult to make it threshold, because now it's at like saying negative 75 millivolts. That's a lot more difference in charge from threshold. All right, any neuron has lots and lots of synapses, lots of synaptic knobs forming synapses on its cell bodies and its dendrites. And so we have this constant stream of information and the release of neurotransmitters and the movement of ions. And again, what's going to be most important is the final migration of all these charges to the axon hillock. So summation is when all of the changes in charge caused by all the movement of these ions reach the axon hillock. If the summation of all those signals lead to threshold being reach negative 55 millivolts to be reached, that will stimulate the voltage-gated channels in the axon hillock, which will lead to the active potential being generated and traveling down the length of the axon. Now, this can be done via what's called spatial summation. Spatial summation just means that, say, for instance, you have lots of different synapses generating depolarization. They all sum up together. They're in different locations, so spatially separated, but the ions eventually make it to the axon hillock, giving us the generation of an axon potential. So lots of different synapses in different areas on the neuron receptive area, leading to the generation of threshold. Temporal summation is when, say, you only have one synapse involved, and it keeps repeatedly signaling for an excitatory response. So it keeps releasing more neurotransmitters, causing more positive voltage-gated ion channels to open, leading to eventually threshold being reached and getting that action potential. So temporal, same place, just over time, keep sending another signal, another signal, another signal, causing lots of depolarization. And then, of course, depolarization leads to the generation of the action potential, which causes the movement of that charge down the length of the axon. So voltage-gated sodium ion channels first open, causing in response to the threshold, and sodium ions flood in, causing massive depolarization, and then they close. And that then causes the next area to get stimulated by that change in charge that causes its voltage-gated sodium ion channels to open, and so on down the length of the axon toward the synaptic knob. So depolarization, boom, 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 causes the sodium ion channels to open and lots of sodium ions flood in, eventually reaching threshold, which then activates the next branch. However, behind it, we have the incorrect distribution of charge, so we don't have all of the sodium ion channels back at their resting state. Many of them are in their inactive state. Repolarization involves the opening of voltage-gated potassium ion channels. They open, potassium ion floods in, leading to a return to resting potential, often with hyperpolarization. It goes even further past the negative 70 millivolts to maybe negative 80 millivolts. But eventually, through the sodium potassium ion pumps, it will return to resting potential. So voltage-gated potassium ion channels open slightly after the voltage-gated sodium ion channels, leading to repolarization. So they're closed, then they open, potassium ions flood out, causing repolarization to occur. Hyperpolarization is because often the voltage-gated potassium ion channels overdo it, and so they have to fix that by the resting membrane potential being reestablished by the sodium-potassium ion pumps. So they make things back to normal.